Welcome to the next session of photosynthesis in higher plants. In the previous session, we came across various experiments that shows that light is necessary for photosynthesis, carbon dioxide is also necessary for the process of photosynthesis, even water is also required for the process of photosynthesis. Now continued with the experiments. So as we came across a heading called as early experiments on the photosynthesis. As I have already told you, there were many scientists, mainly hundreds of scientists were there who were involved in the discovery of the photosynthesis, who were involved in identification of how exactly the photosynthesis occurs. Now we are across the hunting of the photosynthesis, where the elaborate session is discussed in this topic. Then with the continued experiment, let us go to the fourth experiment. Fourth experiment is T.W. Engelmann's experiment. So yesterday we have came across the three experiments. Which shows the photosynthesis occurs in the plants. Now next comes T.W. Engelmann, who has worked from 1843 to from the year 1843 to 1909. The early experiments which we discussed, they were Joseph Priestley experiment, and he was the first person to discover oxygen in the year 1774. Then later on came a person called as John Ingenhousz, now who showed that oxygen bubbles are released during photosynthesis. Almost their experiments were similar, but he confirmed it also. Then later experiment was Julius von Sachs, who showed that glucose is produced during the process of photosynthesis while it is stored in the form of starch in the plants. That is why we say starch is the reserve food material in the plants. Then comes the next experiment, T.W. Engelmann's experiment, and this T.W. Engelmann's experiment is based on a algae called as a cladophora. Now what he does is he takes a prism, then he releases the white light which splits into the spectrum of light. So Vibgyor spectrum. Then what exactly he did? Then what he concluded from these experiments? Okay, so let us see that. Now T.W. Engelmann, what he has did is, he has taken a prism and he released the light. When light is released to the prism, so which already you know, this light, almost we call it as a visible spectra of light, it splits to form into Vibgyor colors. Then after splitting, he has placed a glass plate. A glass plate was taken and he has placed a algae. One algae was placed, and this name of the algae is cladophora. And the suspension was consisting of aerobic microbes. Now this was the suspension which consists of aerobic microbes. Mainly he has taken aerobic bacteria. So let us call it as a bacteria. So he has taken the suspension of aerobic bacteria here. Then look at this part. What was his observation? When light was released to the prism splits the spectra of light into Vibgyor, and this Vibgyor, when it was exposed to a suspension consisting of aerobic microbes in which an algae was placed called as a cladophora, then the wavelength of light, almost at the wavelength of light, red and blue spectra of light, the microbes would accumulate there. Microbes accumulated towards that point because oxygen was releasing from that point. As the microbes were aerobic refers to they respire oxygen, they require oxygen for their survival. These aerobic microbes, they accumulated, they formed into groups at the wavelength of light where there was a red light as well as a blue light. Then he showed that from these two wavelengths of light an absorption spectra as well as action spectra were discovered, and which will be dealt in the later parts of the studies. Okay. Then what he concluded? He concluded that this algae, it works, or maximum wavelength of light that is absorbed by the plants for the evolution of oxygen is red and blue wavelength of light, right. So this is what is called as an Vibgyor spectra of light, right. So we are going to write the statements of this only, right. So what Engelmann has done, T.W. Engelmann, what he has did is he has just exposed the cladophora and algae, and this algae was placed in a suspension consisting of aerobic bacteria. Then he has taken a prism to which the white light was released, where we call it as a visible spectra of light, and it got splitted into Vibgyor, and the oxygen was released at the red wavelength of light as well as the blue wavelength of light, and the microbes got accumulated towards that. That means he showed that for the process of photosynthesis, this visible spectra of light is required for the photosynthesis. Then apart from that, especially the red wavelength and blue wavelength are very much necessary for the process of photosynthesis. This was the conclusion. Okay, so T.W. Engelmann ಮಾಡಿರುವಂಥ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಏನು, ಭಾಳ ಸಿಂಪಲ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡಿದ. ಆಮೇಲೆ ಇದರಿಂದ ಕನ್ಕ್ಲೂಷನ್, ಎ ಲಾರ್ಜ್ ಕನ್ಕ್ಲೂಷನ್ ವಾಸ್ ಡ್ರಾನ್. ಓಕೆ, ಆ ನಂತರದಲ್ಲಿ ಗೊತ್ತಾಯಿತು ಯಾವ ವೇವ್ಲೆಂತ್ ಆಫ್ ಲೈಟ್ ಪ್ಲಾಂಟ್ ಕ್ಲೋರೋಫಿಲ್ ಮಾಲಿಕ್ಯೂಲ್ ಅಬ್ಸಾರ್ವ್ ಮಾಡ್ದದ. ಅದಕ್ಕಾಗಿ ಬೇಸ್ಡ್ ಆನ್ ದಿಸ್ ಈ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮೇಲೆ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಆ್ಯಂಡ್ ಆ್ಯಕ್ಷನ್ ಸ್ಪೆಕ್ಟ್ರಾ. ಅಬ್ಸಾರ್ಪ್ಷನ್ ಅಂತಂದ್ರೆ ಯಾವ ವೇವ್ಲೆಂತ್ ಆಫ್ ಲೈಟ್ ಪ್ಲಾಂಟ್ ಅಬ್ಸಾರ್ವ್ ಮಾಡ್ತಾವ, ದ ಗ್ರೀನ್ ಪಾರ್ಟ್ ಆಫ್ ದ ಪ್ಲಾಂಟ್ ಅಬ್ಸಾರ್ವ್ ಮಾಡ್ದತಿ. ಮತ್ತು ಆ್ಯಕ್ಷನ್ ಅಂತಂತಂದ್ರೆ ಫೋಟೋಸಿಂಥೆಸಿಸ್ ಆ್ಯಕ್ಟಿವಿಟಿ ಹೈ ಇರಬೇಕಂತಂದ್ರೆ ಯಾವ ಸ್ಪೆಕ್ಟ್ರಾ ಬೇಕು ಅಂತ ಅನ್ನೋದು, ವಿಸಿಬಲ್ ಸ್ಪೆಕ್ಟ್ರಾ. ಆ್ಯಂಡ್ ದಿಸ್ ವಿಸಿಬಲ್ ಸ್ಪೆಕ್ಟ್ರಾ ಇಟ್ ರೇಂಜಸ್ ಬಿಟ್ವೀನ್ 400 to 700 nanometer wavelength of light. Okay, so Vibgyor, it lies between 400 to 700 nanometer wavelength of light. We call it as visible spectra. ಇದಕ್ಕೆ ನಾವು ವಿಸಿಬಲ್ ಸ್ಪೆಕ್ಟ್ರಾ ಆಫ್ ಲೈಟ್ ಅಂತ ಕರಿತೀವಿ. ಈ ವಿಸಿಬಲ್ ಸ್ಪೆಕ್ಟ್ರಾ ಒಳಗಡೆ ಈ ವೇವ್ಲೆಂತ್ ಇತ್ತಂತಂದ್ರೆ ಮಾತ್ರ ಪ್ಲಾಂಟ್ ಒಳಗಡೆ ಫೋಟೋಸಿಂಥೆಸಿಸ್ ಆಗ್ತದೆ. Below this and above this wavelength, so it does not help in the process of photosynthesis, right. So this is regarding the part of his experiment, right. Then here what shall we do is, let's write the characters of T.W. Engelmann's. Okay, now look at the characters here, the statement that has been stated after the conclusion of the experiment by T.W. Engelmann. So using a prism, as we have saw the experiment just now, we have written the statements here. Using the prism, he split light, so visible spectra of light, into spectral components and then illuminated a green alga, and the name of that algae is cladophora. Then placed in a suspension of aerobic bacteria. So this was in a Petri plate. Aerobic bacteria were placed. The bacteria were used to detect the sites of oxygen evolution. That means at which wavelength of light the oxygen was evolved. So that was detected by these bacteria. Then he observed that bacteria accumulated mainly in the region of blue and red wavelength of light. So this is most important. So the bacteria got accumulated in those regions where there was evolution of oxygen, and oxygen was evolving at a visible spectra of light where blue and red light was present in this algae, where it used to absorb that wavelength of light of the split spectra. Split means it is splitted into Vibgyor. Then a first action spectra of photosynthesis. So what is this action spectra? What is this absorption spectra? Again we are going to deal it in the next class. So first action spectra of photosynthesis was thus described. So it resembles roughly the absorption spectra of chlorophyll A and chlorophyll B. That means chlorophyll A is the chlorophyll molecule which is present in almost all the plants, along with some other organisms also, where they synthesize their own food, like that of the euglena. Euglena is commonly called as a plant animal. That euglena has a chlorophyll A. But chlorophyll A and chlorophyll B are the pigments which is present in all the plants. Then along with that, chlorophyll A is almost universal in all the plants. Instead of chlorophyll B, chlorophyll C and chlorophyll D can be also present, which you have already studied in the plant kingdom, like that of the algae, green algae, brown algae and red algae, as they have chlorophyll A and B, A and C, A and D. Okay. So, here the absorption spectra of chlorophyll A and chlorophyll B, the chlorophyll molecules are the pigmented molecules. So, they absorb a particular wavelength of light of the visible spectra of the sunlight. So, what absorption spectra or what this visible spectra or which wavelength of light is required for the rate of photosynthesis to increase the process of photosynthesis was first described by T.W. Engelmann. So, regarding the absorption spectra, need not worry here. So, we are going to study with the graph in the next class. Okay. Then almost by the end of 19th century or in the almost in the mid of the 19th century, the equation, the empirical equation for the photosynthesis was given, and with the help of the carbon dioxide, water, so how the carbohydrates were synthesized. So, finally an equation was given where we call it as the empirical formula, and the formula can be written in the form that CO2 plus H2O in presence of light, carbon dioxide and water in presence of light, it produces carbohydrates plus oxygen. So, this was the empirical formula. Now, here CH2O, it refers to a carbohydrate, this refers to a carbohydrate like glucose. Right. So, empirical formula, or we can also call it as the general formula, right. So, by the mid of the 19th century, the equation was framed. Later on, this equation went on changing and the final equation for the photosynthesis was formed. Okay. So, this is regarding the part of the T.W. Engelmann experiment. Now, let us go to the fifth experiment, where we used to call them as early experiments for the process of photosynthesis. Now, to write the characters, to write these characters, just pause the video, rewind it, write the characters. Then the last experiment that we are going to study it now is regarding the Cornelius Van Neel experiment, where he has studied experiment on sulfur bacteria. Okay, so next experiment is regarding Cornelius Van Neel. Okay, so let us call it as the last experiment of Cornelius. Now, this Cornelius, he was present in between 1897 to 1985. Okay, from the year 1897 to 1895. So, what he has stated, right? Now, he has given a milestone experiment just to show that oxygen which is liberated during the process of photosynthesis, it comes from water, not from CO2 during the process of photosynthesis. ದ್ವಿತೀಯ ಸಂಶ್ಲೇಷಣೆ ಕ್ರಿಯೆ ಆಗುವಾಗ ಏನು ಆಕ್ಸಿಜನ್ ರಿಲೀಸ್ ಆಗ್ತದೆ ಪ್ಲಾಂಟ್ಸಿಂದ, ಅದು ವಾಟರ್ದಿಂದ ಬೈ ದ ಬ್ರೇಕ್ ಡೌನ್ ಆಫ್ H2O ದಿಂದ ಬರ್ತದೆ, ಬಟ್ CO2 ದಿಂದ O2 ರಿಲೀಸ್ ಆಗಂಗಿಲ್ಲ ಅಂತನ್ನುವಂಥದ್ದು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಹೇಳ್ತಾ ನಾವು ಬೈ ಯೂಸಿಂಗ್ ಗ್ರೀನ್ ಆ್ಯಂಡ್ ಪರ್ಪಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಇನ್ ಈಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್. ಆ್ಯಂಡ್ ಲೇಟರ್ ಆನ್ ಇನ್ಸ್ಟೆಡ್ ಆಫ್ ವಾಟರ್ ಹೈಡ್ರೋಜನ್ ಡೋನರ್ ಅಂತನ್ನುವಂಥದ್ದು H2S ಹೈಡ್ರೋಜನ್ ಸಲ್ಫೈಡ್ ತೊಗೊಂಡು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡ್ತಾನು. ಆ H2S ದಿಂದ ಲಾಸ್ಟಿಗೆ ಎಂಡ್ ಪ್ರಾಡಕ್ಟ್ ಸಲ್ಫರ್ ಪ್ರಾಡಕ್ಟ್ಸ್ ಬರ್ತಾವೆ. ಈಗ ನಾವು H2O ತೊಗೊಂಡಾಗ ದೆನ್ ದಿಸ್ H2O ಗೆಟ್ಸ್ ಆಕ್ಸಿಡೈಸ್ ಟು ರಿಲೀಸ್ O2. ಆಕ್ಸಿಡೇಷನ್ ಅಂತಂದರೆ ರಿಮೂವಲ್ ಆಫ್ ಹೈಡ್ರೋಜನ್. ಹೈಡ್ರೋಜನ್ ರಿಮೂವ್ ಮಾಡಿದಾಗ O2 ಉಳಿತದ, ಆಕ್ಸಿಜನ್ ರಿಲೀಸ್ ಆಗ್ತತಿ, ಲಿಬ್ರೇಟ್ ಆಗ್ತತಿ ಅಂತ ಹೇಳ್ತಾನೆ ನಾವು. ಸೊ ದಿಸ್ ವಾಸ್ ದ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ವಿಚ್ ವಾಸ್ ಗಿವನ್ ಬೈ Cornelius Van Neel, right. So, what is his experiment? So, let us look at the statement first and then we will discuss what exactly he has given. Okay, so let us look at the statement first. Now, this was the experimental evidence which was given by Cornelius Van Neel. Now, what he stated? He was a microbiologist. So, he was a microbiologist. So, maximum contribution was given by this person only to show that the oxygen that is released during the process of photosynthesis, it comes from water, not from CO2. Now, based on the studies of purple and green bacteria, so he has taken the purple as well as a green colored bacteria in his experiment. So, he demonstrated that photosynthesis is essentially a light dependent reaction, because for the process of photosynthesis light is very much essential and required also. So, in which hydrogen forms a suitable oxidizable compound and reduces carbon dioxide to carbohydrates. That means the hydrogen, as it forms the suitable oxidizable compound, that means from which the hydrogen is removed, finally the H2O gets converted into oxygen. So, when hydrogen is removed, this is called as oxidation. Then later on the carbon dioxide is converted into carbohydrates. So, CO2 is converted into carbohydrates, where it represents the glucose finally stored in the form of starch in the plants. So, here an equation follows where 2H2A plus CO2 in presence of light, it forms into 2A plus CH2O, which refers to a compound called as carbohydrate plus water. Now, here there is what hydrogen, when hydrogen is withdrawn, that means removed, oxygen is released. So, here A refers to, this is nothing but this is oxygen only. So, 2H2A means if it is H2O, so here 2 molecules of oxygen is released. Okay, so O2 is released during the process of photosynthesis. That was the equation which was stated by this person. Then in green plants, H2O is, this is most important, H2O is the hydrogen donor for purple and green sulfur bacteria. So, this is in the green plants. H2O is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur and not oxygen. Suppose if the gas like H2S, hydrogen sulfide, is used instead of H2O. Now, here in green plants, H2O is the hydrogen donor for purple as well as the green sulfur bacteria. Now, instead of water, so let us change the statement here. So, in the bacteria, now if H2S is used, hydrogen sulfide, if H2S is used, the oxidation product is a sulfur and it is not oxygen, because H2S. So, here add the statement instead of water. So, insert the statement here. Instead of water, if H2S is the product that is used, that means if H2S is the oxidation product, oxidation means removal of hydrogen, then the product would be sulfur and not oxygen. In case of H2S, if the product hydrogen sulfide is used instead of water, then the product is sulfur. H2O use madidondre product is oxygen. That was the statement which was drawn by Neel. Then he inferred that oxygen evolved by green plant comes from water and not from CO2 by using H2S, hydrogen sulfide use ಮಾಡಿಕೊಂಡು ಅವೇನು ಕಂಡುಕೊಳ್ತಾನಂತಂದ್ರೆ ಅದರಿಂದ ಪ್ರಾಡಕ್ಟ್ ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಒಳಗಡೆ ಏನು ಬರ್ತಾ ಇತ್ತು ಅಂತಂದ್ರೆ ಗ್ರೀನ್ ಆ್ಯಂಡ್ ಪರ್ಪಲ್ ಸಲ್ಫರ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಎಂಡ್ ಪ್ರಾಡಕ್ಟ್ ಸಲ್ಫರ್ ಬರ್ತಾ ಇತ್ತು, ಆಕ್ಸಿಜನ್ ಅಲ್ಲ. ಬಟ್ H2O ಯೂಸ್ ಮಾಡಿದಾಗ ಎಂಡ್ ಪ್ರಾಡಕ್ಟ್ ಆಕ್ಸಿಜನ್ ಬರ್ತದ. ದ್ಯಾಟ್ ಮೀನ್ಸ್ ಆಕ್ಸಿಜನ್ ವಿಚ್ ಇಸ್ ಲಿಬರೇಟೆಡ್ ಆರ್ ಇವಾಲ್ವ್ ಡ್ಯೂರಿಂಗ್ ಫೋಟೋಸಿಂಥೆಸಿಸ್ ಇಟ್ ಕಮ್ಸ್ ಫ್ರಮ್ ವಾಟರ್, ನಾಟ್ ಫ್ರಮ್ CO2, ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡಿಂದ ಅಲ್ಲ. ಯಾಕಂದರೆ ಭಾಳಷ್ಟು ಜನ ಇದ್ರ ಮೇಲೆ ಕಾಂಟ್ರವರ್ಷಿಯಲ್ ಇತ್ತು. ಆಕ್ಸಿಜನ್ ಏನು ರಿಲೀಸ್ ಆಗ್ತೈತೆ ಅದು CO2ದಿಂದ ಕಾರ್ಬನ್ ಇದ್ದದ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್ ಆಗ್ತೈತಿ, O2 ಇದ್ದದ ರಿಲೀಸ್ ಆಗ್ತೈತಿ ಅಂತ ಹೇಳ್ತಾ ಇದ್ರು. ಬಟ್ ಇವು ಎ ಮೈಲ್ ಸ್ಟೋನ್ ಕಾಂಟ್ರಿಬ್ಯೂಷನ್, ಇವು ಮೇಜರ್ ಕಾಂಟ್ರಿಬ್ಯೂಷನ್ ಇತ್ತು ಅವನ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ದಿಂದ, ಆಸ್ ಹಿ ವಾಸ್ ಎ ಮೈಕ್ರೋಬಯಾಲಜಿಸ್ಟ್ ಹೂ ವರ್ಕ್ಡ್ ಆನ್ ದೀಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್. ಓಕೆ, then later on by using the radioactive isotopes also it was proved that the oxygen which is released during photosynthesis comes from what? Water, not from CO2 antanuvantada. By using radioactive isotopes also it was confirmed. Okay, so that was the later research. So this was later proven by using radioactive isotopes. ಓಕೆ, ಆ ನಂತರದಲ್ಲಿ ರೇಡಿಯೋಆಕ್ಟಿವ್ ಐಸೋಟೋಪ್ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಈ ಆಕ್ಸಿಜನ್ ರಿಲೀಸ್ ಏನಾಗ್ತದೆ, ಇದು ಎಲ್ಲಿಂದ ರಿಲೀಸ್ ಆಗ್ತದೆ, ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡು ವಾಟರ್ ಅಂತ ಅನ್ನುವಂಥದ್ದು ಎಕ್ಸ್ಪೆರಿಮೆಂಟಲ್ ಎವಿಡೆನ್ಸ್ ಮುಖಾಂತರ ಪ್ರೂವ್ ಮಾಡ್ತಾರೆ. ದೆನ್ ಫೈನಲಿ an equation for photosynthesis, a final equation for photosynthesis was described, and that final equation comes to be 6CO2 plus 12H2O in presence of sunlight and bearing chlorophyll ಇದ್ದಾಗ ಮಾತ್ರ ಈ ರೀತಿ ಆಗುವಂಥದ್ದು. Then the product that we get is C6H12O6 plus 6H2O plus 6O2. So this was the final equation for the photosynthesis. So look at the equation here. So this equation was given finally. Now usually photosynthesis definition ಕೊಟ್ಬಿಟ್ಟು equation for the photosynthesis ಅಂತ directly ಇಂದ ಬರಿತೀವಿ, but ಈ equation ಬರಿಬೇಕಂತಂದ್ರೆ there were series of experiments which were carried out by hundreds of scientists who worked on the process of photosynthesis, and the final equation for the photosynthesis was drawn. And here it goes with if 6 molecules of carbon dioxide and 12 molecules of water in presence of sunlight and chlorophyll, it produces 1 molecule of glucose, C6H12O6 is a glucose, and 6 molecules of water is released and 6 molecules of oxygen is released. So this is regarding the final equation for the process of photosynthesis. Okay, so what you need to do is just pause the video and write all the characters, right? So with this, let's move on to the next part. Next part is, as we know, for the process of photosynthesis occurs in which structure of the plant? Photosynthesis generally occurs in the leaf part. Okay, now leaf is a flattened structure where it is having a dorsal surface and a ventral surface. It is dorso-ventrally flattened structure. Upper part we call it as upper epidermis, lower part we call it as lower epidermis. In between these two epidermis, leaf flat irtada. ಎರಡು ಎಪಿಡರ್ಮಿಸ್ ಮಧ್ಯೆ ಇರುವಂಥ ರೀಜನ್ ಮೀಸೋಫಿಲ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಇನ್ ದ ಮೀಸೋಫಿಲ್, ಫೋಟೋಸಿಂಥೆಸಿಸ್ ಆಕರ್ಸ್ ಇನ್ ದ ಮೀಸೋಫಿಲ್ ರೀಜನ್ ಆಫ್ ದ ಲೀಫ್ ಅಂತ ಕರಿತೀವಿ. ಸಪೋಸ್ ಇಫ್ ವಿ ಟೇಕ್ ದ ಲೀಫ್ ನಾವು ಇಲ್ಲಿ ಈ ರೀತಿ ತೊಗೊಂಡಾಗ ಇದು ಅಪ್ಪರ್ ಎಪಿಡರ್ಮಿಸ, ಇದು ಲೋವರ್ ಎಪಿಡರ್ಮಿಸ, ಈ ಮಧ್ಯದಲ್ಲಿ ರೀಜನ್ಸ ಇದು ಮೀಸೋಫಿಲ್ ರೀಜನ್ ಅಂತ ಕರಿತೀವಿ. ಈ ಮೀಸೋಫಿಲ್ ರೀಜನ್ಸ್ ಒಳಗಡೆ ಸೆಲ್ಸ್ ಇರ್ತಾವೆ. ಆ ಸೆಲ್ಸ್ ಯಾವ ಅಂತಂದ್ರೆ ಅವು ಪ್ಯಾರೆಂಕಮಾ ಸೆಲ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಆ ಪ್ಯಾರೆಂಕಮಾ ಸೆಲ್ಸ್ ಒಳಗಡೆ ಇದರೊಳಗಡೆ ಇಲ್ಲಿ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ಗಳು ಇರ್ತಾವೆ, ಹಲವಾರು ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ಗಳು ಇರ್ತಾವೆ. ದ ಪ್ಯಾರೆಂಕಮಾ ಸೆಲ್ಸ್ ಹ್ಯಾವಿಂಗ್ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್, ಆ ಪ್ಯಾರೆಂಕಮಾ ಸೆಲ್ಸ್ಗೆ ಕ್ಲೋರಂಕೈಮಾ ಅಂತ ಕರೀತಾರೆ. ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಎಸ್ ಅ ಕ್ಲೋರಂಕೈಮಾ. ದ ಪ್ಯಾರೆಂಕಮಾ ಸೆಲ್ಸ್ ಹ್ಯಾವಿಂಗ್ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್, ದೇ ಆರ್ ಕಾಲ್ಡ್ ಎಸ್ ಅ ಕ್ಲೋರಂಕೈಮಾ. ದೆನ್ ದ ರೀಜನ್, ಈ ಮಧ್ಯದಲ್ಲಿರುವಂಥ ರೀಜನ್, ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಎಸ್ ಅ ಮೀಸೋಫಿಲ್. This is mesophil. Upper part, this is called as a upper epidermis. Lower part, this is called as a lower epidermis. Upper epidermis, lower epidermis ಅಂತ ಕರಿತೀವಿ. ಮಧ್ಯದಲ್ಲಿ ಇರುವಂಥ ರೀಜನ್, this is called as a mesophil region. This mesophil region consists of parenchyma cells, and inside the parenchyma cells they consist of chloroplast. ಅದರ ಒಳಗಡೆ chloroplast ಇರ್ತಾವೆ. Right, now we are moving across the structure of the chloroplast, and already regarding the structure of the chloroplast you have studied it in the cell chapter, and the same thing is repeated here. We are representing the same diagram because the next process everything occurs in the chloroplast. So that's why we should know the structure of the chloroplast, and hence the structure of the chloroplast is again repeated in this chapter, as already you have studied in the cell chapter. So already we have came across the structure of the chloroplast, we have described the entire things of the chloroplast. Now suppose if we look at the structure of the chloroplast. Now chloroplast is a again double membrane structure. So here I am drawing a 2D diagram here, where the 3D diagram is shown in the cell chapter, chapter number 8. Okay. Now inside this we have chloroplast. This is the structure of a chloroplast. So here it is how it looks. Okay. So this is regarding the granum that we have already discussed, and the same thing is also described here. Okay. And in between we have certain granular structures where we call them as starch granules, and certain lipid droplets, certain ribosomes are also there. These are the ribosomes. Now this is the outer membrane, below is the inner membrane, and inside this is the stroma. This entire structure is called as a single is a granum, both together grana, that is a plural term, and each single unit of this is called as a thylakoid. This is thylakoid, and this is the lipid droplet. This is the lipid droplet. These are starch granules. Okay, so these are the starch granules. So this is regarding the structure of a chloroplast, which already we know. Now look at this. This chloroplast is present here. You one single unit ಏನು ಕಾಣಿಸ್ತಾವೆ dots ಇವ chloroplast ಅಂತ ಕರಿತೀವಿ. So chloroplast appears in this way. Chloroplast is a double membrane structure which is present in the mesophyll region of the leaves. Leaves ಒಳಗಡೆ ಇರ್ತಾವೆ. Their main function is photosynthesis. Photosynthesis exactly ಆಗೋದು ಇಲ್ಲೇ. Okay, one is in the thylakoid ಒಳಗಡೆ ಆಗ್ತದ, ಆಮೇಲೆ ಸೆಕೆಂಡ್ ಪಾರ್ಟ್ ಸ್ಟ್ರೋಮಾದ ಆಗ್ತದ. ಇವು ಎರಡು ರೀಜನ್. The next chapter, major part that we are going to discuss is in the part of the thylakoid as well as in the stroma. So until and unless we complete the chapter, you just be on the chloroplast. ಈಗ ಸಮಯ ಏನಂತಂದ್ರೆ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಮೇಲೆ ಹತ್ತಿ ನಿಂತು ಇಲ್ಲಿ ಇದ್ರೊಳಗಡೆ ಏನೇನು ಆಗ್ತೈತಿ ಅನ್ನೋಂಥ ಎಲ್ಲ ಪ್ರೋಸೆಸ್ ನೋಡೋಣ. ಓಕೆ, ಸೊ ನೆಕ್ಸ್ಟ್ ಟಿಲ್ ದ ಎಂಡ್ ಆಫ್ ದ ಚಾಪ್ಟರ್ ಯು ಆರ್ ಇನ್ ದ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್. ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಬಿಟ್ಟು ಹೊರಗಡೆ ಬರಬೇಡ್ರಿ, ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಒಳಗಡೆನೇ ಇರಿ. ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಒಳಗಡೆ ಏನೇನು ಆಗ್ತೈತಿ ಅನ್ನೋಂಥ ರಿಯಾಕ್ಷನ್ಸ್ ಬಗ್ಗೆನೇ ಸ್ಟಡಿ ಮಾಡೋಣ. ಓಕೆ, ಸೊ till the end of the chapter we are at the part of the chloroplast only, because the detailed part, the elaborate part, the entire discussion part we are discussing with respect to the chloroplast only, right? Then here along with that what we have, idr jotege matte illi ಏನು ಇರೋದು, along with that we have the lipid droplets. Certain ribosomes are there, because ribosomes are the cell organelles where we call them as non-membrane bond cell organelles which are present in the chloroplast also. Then it has a certain starch granules. ಇಲ್ಲಿ ಕಾಣಿಸುವಂಥವು ಚಕ್ಕಲಿ ಅಲ್ಲ, ಅವು starch granules. Okay, so starch is the reserve food material in the plants. Now the membrane of the thylakoid, it produces the energy in the form of ATP as well as NADPH. ಈ thylakoid ದಾಗ ಪ್ರೊಡ್ಯೂಸ್ ಆಗೋದು ATP and NADP. These are energy source compounds. Okay, so NADP is a nicotinamide adenine dinucleotide phosphate and ಕರಿತೀವಿ. ATP and NADP are produced in presence of light. So that's why the reactions that occurs in the membrane of the thylakoid, we call them as light reactions because they are light dependent reactions. And the reactions that occurs in the stroma, they are light independent reactions. That's why the reactions which occurs in the stroma, they are said to be dark reactions. These are said to be dark reactions. And the reactions that occurs in the thylakoid, they are said to be light reactions. Light reactions means which takes place in presence of sunlight. ಅದರ ಬಗ್ಗೆ ನಿಮ್ಗೆ ಅರ್ಥ ಆಗ್ತದೆ. ಫೋಟೋಸಿಂಥೆಸಿಸ್ ಲೈಟ್ ಬೇಕಲ್ಲ, ಲೈಟ್ ಇದ್ದಾಗ ಥೈಲಾಕೋಡ್ ಒಳಗಡೆ ಮೆಂಬ್ರೇನ್ ಇರ್ತೈತಿ. ಇಟ್ ಇಸ್ ಆಲ್ಸೋ ಎ ಡಬಲ್ ಮೆಂಬ್ರೇನ್ ಬಾಂಡ್ ಸ್ಟ್ರಕ್ಚರ್. ಆನ್ ದ ಮೆಂಬ್ರೇನ್ ಆಫ್ ದ ಥೈಲಾಕೋಡ್, ಇಟ್ ಪ್ರೊಡ್ಯೂಸಸ್ ಎನರ್ಜಿ ಲೈಕ್ ATP and NADP in presence of sunlight. That's why the reactions which occurs in the membrane of the thylakoid are said to be light reactions. While in the stroma, there is an enzymatic process where a cycle occurs. Illon cycle ಆಗ್ತದ by which the food is produced, and those reactions are said to be dark reactions. Dark reaction doesn't mean that it takes place in night time. ಇದು ರಾತ್ರಿ ಹೊತ್ತು ಆಗಂಗಿಲ್ಲ. ಇದು ಡೇ ಟೈಮ್ ಒಳಗಡೆ ಥೈಲಾಕೋಡ್ ಏನು ಎನರ್ಜಿ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರ್ತೈತಿ, ಈ ಎನರ್ಜಿ ಯುಟಿಲೈಸ್ ಮಾಡಿಕೊಂಡು ಫುಡ್ ತಯಾರು ಮಾಡ್ತೈತಿ ಇನ್ ದ ಫಾರ್ಮ್ ಆಫ್ ಸ್ಟಾರ್ಚ್. ಬಟ್ ಇದಕ್ಕೆ ಎನರ್ಜಿ ಬೇಕು. ಎನರ್ಜಿ ಇಲ್ಲ, ATP NADP ಇಲ್ಲ ಅಂತಂದ್ರೆ ಫುಡ್ ತಯಾರಾಗಂಗಿಲ್ಲ. ಹೆಂಗೆ ಆಗಂಗಿಲ್ಲ, ಇದ್ರ ಡಿಟೇಲ್ ಅಕೌಂಟ್ ನೆಕ್ಸ್ಟ್ ಪೀರಿಯಡ್ ಒಳಗಡೆ ಬರ್ತದೆ. ಓಕೆ, ಸೊ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಡಿಸ್ಕಸ್ ಇಟ್ ಇನ್ ಎಲಾಬ್ರೇಟ್ ಪಾರ್ಟ್, ರೈಟ್. ಜಸ್ಟ್ ಸಿಂಪ್ಲಿ ರಿಮೆಂಬರ್ ದ್ಯಾಟ್ ಡಾರ್ಕ್ ರಿಯಾಕ್ಷನ್ ಈಸ್ ಡಿಪೆಂಡೆಂಟ್ ಆನ್ ಲೈಟ್ ರಿಯಾಕ್ಷನ್. ಲೈಟ್ ರಿಯಾಕ್ಷನ್ ಆದ ನಂತರ ಡಾರ್ಕ್ ಆಗ್ತದೆ, ಡಾರ್ಕ್ ಆದ ನಂತರ ಲೈಟ್ ಅಲ್ಲ. ಯಾಕಂತಂದ್ರೆ ಈ ಎನರ್ಜಿ ಯೂಸ್ ಮಾಡ್ಕೊಂಡು ಇಲ್ಲಿ ಫುಡ್ ತಯಾರು ಮಾಡ್ತೈತಿ. ಅಂಡರ್ಸ್ಟುಡ್? ಈ ಎನರ್ಜಿ, ದ ಎನರ್ಜಿ ವಿಚ್ ಇಸ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ in the presence of sunlight in the form of ATP and NADPH are used in the stroma, and there is a enzymatic process. ಅಲ್ಲೊಂದು ಸೈಕಲ್ ಆಗ್ತದೆ. ಆ ಸೈಕಲ್ ನಾವು ಕೆಲ್ವಿನ್ ಸೈಕಲ್ ಅಂತ ಕರಿತೀವಿ. ಏನದು ಕೆಲ್ವಿನ್ ಸೈಕಲ್? ಇಟ್ಸ್ ಅ ವೆರಿ ಬಿಗ್ ಸೈಕಲ್ ವಿಚ್ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಸ್ಟಡಿ ಇಟ್ ಇನ್ upcoming class. Just simply remember, in presence of light, the reactions which occurs in the thylakoid, we call them as light reactions. And in presence or absence of light ಇರಲಿ ಬಿಡಲಿ, ಎನರ್ಜಿ ಇತ್ತಂದ್ರೆ ಡಾರ್ಕ್ ರಿಯಾಕ್ಷನ್ಸ್ ಆಗ್ತಾ ಇರ್ತಾವ. ಡಾರ್ಕ್ ರಿಯಾಕ್ಷನ್ ಡಸ್ ನಾಟ್ ಮೀನ್ ದಟ್ ಇಟ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್ ಇನ್ ನೈಟ್ ಟೈಮ್. ರಾತ್ರಿ ಹೊತ್ತು ಆಗಂಗಿಲ್ಲ. ಡಾರ್ಕ್ ಅಂತ ಯಾಕೆ ಕರೀತಾರೆ ಅಂದ್ರೆ ಅದು ಲೈಟ್ ಇಂಡಿಪೆಂಡೆಂಟ್ ರಿಯಾಕ್ಷನ್. ಇಟ್ ಈಸ್ ನಾಟ್ ಡೈರೆಕ್ಟ್ಲಿ ಡಿಪೆಂಡೆಂಟ್ ಆನ್ ಲೈಟ್, ಬಟ್ ಇಟ್ ಈಸ್ ಇಂಡೈರೆಕ್ಟ್ಲಿ ಡಿಪೆಂಡೆಂಟ್ ಆನ್ ಲೈಟ್. ಇದು ಹೆಂಗೆ ಅಂತಂದ್ರೆ, ನಾವು ಹೆಂಗೆ ಲೈಟ್ ಮೇಲೆ ಡೈರೆಕ್ಟ್ಲಿ ಡಿಪೆಂಡ್ ಇಲ್ಲ, ಬಟ್ ಇಂಡೈರೆಕ್ಟ್ಲಿ ಹಂಗೆ, ಬಿಕಾಸ್ ವಿ ಆರ್ ಡಿಪೆಂಡ್ ಆನ್ ದ ಪ್ಲಾಂಟ್ಸ್ ಫಾರ್ ಅವರ್ ಫುಡ್, for the source of food, right? So remember this structure, and based on the structure, next part we are going to deal in detail account. So till then, stay tuned. Thank you.